CRISPR technology has revolutionized genetic engineering in the past 10 years. The most frequently used form of CRISPR works by using an enzyme called Cas9, which is an endonuclease, meaning that it can cut DNA inside a living cell. To guide the enzyme to the right location on the cell's genome, a small piece of synthetically produced RNA, called guide RNA, can be inserted into Cas9. The guide RNA contains the complementary code for the target sequence on the genome you want to cut. So this means that CRISPR technology is a pair of genetic scissors that can cut DNA exactly where you want. The reason to cut open the DNA is that open DNA can be changed. One possibility is to introduce a new sequence of DNA into the cell. This is called a donor sequence. The cell's own repair mechanisms will then incorporate the donor sequence at the place where the DNA was cut by Cas9. The new sequence may code for properties that can potentially cure various genetic disorders. However, CRISPR technology is limited by the fact that only relatively small donor sequences of a few hundred base pairs in size can be inserted into the genome. The next genetic tool, in the form of a CRISPR-associated transposon, also called CAST, can do some of what CRISPR-Cas9 lacks. In the CAST system, Cas enzymes work together with a transposon that is used to carry larger pieces of DNA. This is capable of inserting pieces of DNA up to 30,000 base pairs in size.
It allows us to insert not just parts of genes or small genes, but multiple genes and also regulatory regions to control the gene expression.
The finer details as to how the DNA is inserted are not yet fully understood, but now researchers have characterized the structure of the transposon part of CAST. And this paves the way for further developing the CRISPR system towards use in both research and clinical practice.
We have both characterized the structure of the transposon and introduced genetic changes so that we can increase the speed and precision of its ability to insert large pieces of donor DNA into the genome. It shows that it's possible to change the system and optimize it.
All of these are steps towards being able to use the technology on human cells at some point in the future. Once researchers develop the tool to be used in humans, the possibilities are almost endless. You can read more about this research in the article at sciencenews.dk.